in total, and the first one in the year 2022, Amplifying Voices webinar, the Early Career Researchers Making Waves in Aquatic Sciences. The Amplifying Voices webinar is a new initiative. We started this in November 2021 by ASLO and specifically by the Early Career Committee. My name is Mina and I will moderate session today, and I'm one of the members of Early Career Committee. And also today on the call there are other members, a few other members here. Through our work and the support of ASLO board and the ASLO communications office, we started the seminar series to highlight and particularly to amplify the voices of early career aquatic scientists from across the world. And here specifically those from historically excluded groups, and this includes ethnicity and ethnical origin, disability, sex, gender, sexual orientation, or any other sort of identity, or any other sort of, or excluded identity or groups. So far, we had two successful sessions in 2021 in November and December. And if you missed that out, please have a look and watch them on YouTube after. We're very excited today. We have two speakers, one from Sweden and one from Japan. But before I begin with introducing them, I would like to highlight that we have scheduled talks at the end of February and also March. And if you are an early career researcher in aquatic sciences and interested in applying to be a speaker, please send your application or contact us. You can send your email to the address that is posted on this slide. Contact us at Early Career Committee. We would really love to hear from you and take your research out to a wider audience. Our first speaker for today is Dr. Justyna Hampel. She's currently a postdoctoral researcher at Stockholm University in Sweden, and she's working on microbial interactions between cyanobacteria and heterotrophs. She received her PhD in environmental sciences at Wright State University in Archaea, US, where she studied nitrogen cycling in eutrophic lakes. After her PhD, in 2019, she worked as a postdoc at the University of South Mississippi, and she looked there at the impacts of artificial habitats on deep sea microbial communities in the Gulf of Mexico. And she's going to talk to us today about how distinct microbiomes flourish around sunken ships as they become artificial reefs. I'm very excited to hear more about it. Our second speaker for today is Dr. Kuo Hong Wong, Josh Wong, and he's going to talk about zinc silicate decoupling in the subarctic Pacific. And this will be very interesting to see, since zinc is vital for phytoplankton, for physiology of oceanic phytoplankton. So Josh did his bachelor in Cambridge, and from there he continued to University of Tokyo in Japan, where he completed his master's and PhD and continued for postdoc position. He's now an assistant professor at Kanazawa University in Japan. In 21, Josh received Young Researchers Award in Marine Chemistry from the Research Institute for Oceanography. His work focused mainly on distribution and processes that are related to metal ions in the water. Now we will have 20 minutes presentations by Justyna, and then followed by 5 to 10 minutes discussion. Then Josh will present, and then we will have an overall discussion about, we will have first discussion on his talk, and then we will have overall discussion with both speakers at the end. If you have any questions during the talks, please just post them in the chat or wait till the talk is over, and then we can take them. So with this, we are ready to welcome Justyna and learn more about her research. And
hi, my name is Justyna Hampel, and I am currently a postdoc at Stockholm University in Sweden. And today I'll be presenting some work.
I think there's a problem with the slides. Sorry, I don't see any of the slides. Is that everybody's experience?
So, okay, thank you, Roxanne. I will then try again. Can you see a slide now?
No, I think you need to switch which window you're sharing. We still see your PowerPoint slides.
I don't see any slides at all.
And now? Okay, so I will first unshare and then I will share it again. Maybe that will work.
Yeah.
I apologize for this. I just forgot to mention that we will play Justyna's pre-recorded talk. She is here with us in the audience, but she will not be able to give us talk because of bad connection, but she will be here for discussion round. And now I'm going to share it again. Can you all see it now?
Yes.
Yeah.
Very good. Yes, I just need to play it.
Hi, my name is Justyna Hampel, and I am currently a postdoc at Stockholm University in Sweden. And today I'll be presenting some work from my previous postdoctoral position at University of Southern Mississippi with Dr. Leila Hamdan, my postdoc advisor, and Rachel Mosley, a graduate student in the lab. So the title of this talk is Microbiomes Respond Predictably to Build Habitats on the Seafloor. And I just wanted to take a moment to
We lost the sound, Mina. Sorry.
Yeah, Mina, I don't think you can mute yourself. There we go.
Respond predictably to build habitats on the seafloor. And I just wanted to take a moment to quickly acknowledge and thank the ASLO Early Career Committee for giving me a chance to present this research. The ocean seafloor is the largest continuous ecosystem on Earth. However, less than 20% of the seabed has been mapped so far. The advances in high-resolution bathymetry report high abundance of geological features on the seafloor, such as hydrothermal vents, cold seeps, gas hydrates, canyons, and smaller habitat features, all of which contribute to heterogeneous landscape of the seabed. So it's not this barren, flat surface under the water. This habitat heterogeneity is thought to be a major factor influencing biodiversity on the seabed. So the habitat heterogeneity allows for more species to coexist in a specific area, increases species diversity and richness, and leads to a greater number of ecological niches. Geological habitat features differ quite a bit from the surrounding soft sediment seabed. And these include hydrothermal vents and cold seeps and methane hydrates, where the temperature and strong chemical gradients select for very specific populations and harbor genetically distinct microbes. Organic falls, such as whale carcasses and wood falls, also create a unique environment in the surrounding sediments, where, for example, decomposition of the organic matter creates steep redox gradients in sediments and attracts diverse microbial life. And even recent studies mention that things like rock patches in abyssal plains can contribute to biodiversity and sort of act as a hotspot of biodiversity on the seabed. So we see that these natural habitats influence diversity of microbes, but also macrofauna on the seafloor. In comparison to geological and biological features on the seabed, built environments on the seafloor are relatively understudied. And these built environments are also distinct from the surrounding sediments and provide hard substrate surfaces that allow for biofilm formation or biofouling that can later lead to development of artificial reefs. And built environments can include anything from oil and gas infrastructure, which the extent of you can see here on this map in the Gulf of Mexico with all the oil rigs and pipelines. And they can also include shipwrecks and airplane wrecks, sunken ammunition, and barrels of toxic waste like the ones from off the shore of Catalina Island in California. In this study, we used shipwrecks as a model system of built habitat on the seafloor to investigate how these built features or unnatural structures on the seabed influence the surrounding environment. We know from macrofaunal studies that shipwrecks indeed transform into artificial reefs and have really positive influence of microbiological diversity. Shipwrecks are also widespread across the global seafloor. UNESCO estimates over 3 million shipwrecks on the global seafloor. And we know there's over 2,000 known historic shipwrecks in the Gulf of Mexico that you can see on this map, and over 100,000 known shipwrecks in the Baltic Sea. So there's some places in the ocean where these structures are in high abundance and high density on the seabed. While they have positive influence on macrofaunal biodiversity, there's also some environmental hazards to consider. For example, some shipwrecks can contain sunken munition or chemical weapons and toxic chemicals that can leak onto the surrounding seafloor. And also, if they are in high density on the seafloor, they can serve as potential stepping stones for invasive species. So we don't fully understand their entire influence on the seafloor ecosystem in general. So while artificial reefs have been studied for their influence of macrofauna communities, things like fisheries, they have largely been overlooked when it comes to microbial studies. And the metadata analysis that I'm gonna talk about today kind of builds off of previous work in the Hamden lab, where we started noticing patterns of microbiological response to these built habitats on the seafloor. So in the pilot sort of study, we investigated the influence of metal-hulled shipwreck Annona in the Gulf of Mexico, and we found increased bacterial richness and diversity as a function of proximity to the shipwreck. So here on these graphs, you can see we plotted Shannon diversity around the shipwreck from an aerial view and also from a cross-section of the sediment, with the red color being higher diversity. And we observed this sort of island effect surrounding the shipwreck, where the diversity was highest around the shipwreck and it extended 200 meters away from the shipwreck. We also observed a halo of unique core taxa that also extended to the same distance. And that sort of led to the hypothesis that shipwrecks represent island-like systems for marine microbes in sediments. In another study, we focused on 2 wooden shipwrecks in the Gulf of Mexico at different depths. One was at 500 meters, the other one was 1,800 meters. And we found increased diversity of Archaea, specifically in sediments around the shipwreck, and changes in the community structure across a 60-meter transect extending away from the shipwreck. What was interesting also was that the changes were more pronounced at the deeper side at 1,800 meters, suggesting that far from coastal influence and terrestrial input, these shipwrecks might be significant sources of organic matter in sort of more oligotrophic ocean settings. And also because they're made out of wood, we think that there might be some high organic matter decomposition rate happening. So as you can see, the deep sea is not free of marine debris and marine construction, especially in the coastal zones, but also in the very deep ocean. And we observe rising number of built features on the seafloor. For example, 60% of offshore energy platforms are located in the Gulf of Mexico. And this is something that we call an ocean sprawl, or increasing influence of anthropogenic construction that extends from land into the sea. And there's studies that show that there's going to be a predicted increase of 50 to 70% in global marine infrastructure by 2028. And the sphere of influence, or how these built features affect the seafloor ecosystems and the dynamics between species, is very poorly understood. And the response of microbial communities, specifically to build habitats, is particularly interesting since microorganisms are the primary colonizers of substrate and hard surfaces in the ocean. So the objectives of this meta-analysis study was to determine how historic deep sea shipwrecks of different size, age, and hull construction shape microbiome composition in the surrounding sediments. And we also wanted to determine whether seafloor microbes respond to these built features or built habitat in a stochastic or predictable manner. So in the span of 5 years, we collected this massive database of sediment cores surrounding 9 historic shipwrecks in the Gulf of Mexico. So just to orient you here, these sites are south of the Mississippi and Louisiana coastline in the northern Gulf of Mexico. And our sites included 3 shipwrecks that were steel-hulled. That was Alcoa, Puritan, Annona, and Halo, and 6 wooden shipwrecks. And all the shipwrecks ranged in depth between 144 meters, which is the shallowest one, Halo here, to almost 2,000 meters, which is this huge shipwreck, Alcoa, Puritan, deep in the Gulf of Mexico. So just really quickly to take you through the methods, sample collection spanned over 8 offshore expeditions between 2014 and 2019 aboard RV Pointer and Pelican in the Gulf of Mexico. We collected sediment cores using the remotely operated vehicles Global Explorer and Odysseus, based on which cruise we went to, with Jason-style push cores that you can see in this picture here. And at sites that were farther from the shipwreck, or at distances that were further from the shipwreck, we also used multi-core to collect these sediment samples. So overall, we collected 666 archaeal samples and 788 bacterial samples around these 9 historic shipwrecks. And like I said, they ranged quite a bit in depth. We sampled along either a single or multiple 4, usually radial transects extending away from the shipwreck, where closest sites or closest samples were collected 2 meters away. And the furthest distance that we collected was 1,000 meters away from the shipwreck. And usually our core depth span between 2, or the surface top layer of the core, up to 18 centimeters deep. So because we were primarily interested in looking at microbial diversity in these sediments and the changes in the community structure, we used 16S rRNA amplicon sequencing on the extracted DNA from the sediment samples on the aluminum MySeq platform. And then we followed with downstream analysis of amplicons in QIIME2, where we used the data2 plugin for quality control and creation of the ASV table. And we used research plugin with Silva database for taxonomic assignment. We also did a core microbiome analysis in these samples. And lastly, because we were trying to answer the question of the predictability of a microbial community around built environments, we used the QIIME2 sample classifier supervised learning plugin with random forest, where we were interested to look at the prediction of sample metadata. In this case, it was the distance from shipwrecks based on specific ASVs present in these samples. And we also looked at the similarity matrix, Baker's dissimilarity matrix in primary, and we used Permanova to identify differences in the community composition based on the distance from the shipwreck, the sediment depth, the water depth, and also the hull type of the shipwreck. I'm going to begin with showing you the changes in Shannon diversity with distance going away from the shipwrecks. And again, this meta-analysis includes all 9 shipwrecks in these plots, but the patterns we observed in the compiled data set of all shipwrecks were also present at individual sites. So here on this panel, on the top portion of it, you can see the decline in Shannon index or Shannon diversity with distance going away from the shipwreck. And on the left here, this is the data for Archaea, and on the right for bacteria. So in both cases, we observed a decrease in diversity as we went away from the shipwreck from the 2-meter distance to 1,000 meters. And this was much more pronounced in the Archaea samples, but also significant pattern in the bacteria. And on the bottom of the panel, you can see the spatial analysis of archaeal and bacterial diversity with distance. Contour plots were constructed from the data from all 4 transects and aggregated on one axis. And the grid was created with Kriging method of extrapolation. So again, on the left, Archaea, on the right, bacteria. Overall, we observed higher diversity in the bacteria than in Archaea, but in both cases, again, we see this pattern of highest diversity in the first 200 meters away from the shipwreck, or 250 meters away from the shipwreck. And after that distance, the diversity sort of trails off in both archaeal and bacterial samples. So next we're looking at archaeal core microbiome surrounding the shipwrecks sampled in this study. So our parameter for the core microbiome was 80%. So now we are looking at the ASVs present in 80% of all the collected samples, which for Archaea was 532 samples in these distance groups of between 2 and 1,000 meters. And the size of the bubble indicates the relative abundance or the contribution of specific ASV to the core microbiome. So right away, you can see that there is this increased diversity and more various members of the core microbiome between the 2 and 150 meters. And after that, the only contribution, the main contribution to the core microbiome was Tamarchiota. But in this specific distance between 2 and 150 meters, we have high abundance of Archaea, like Bathyarchaea or Lochiarchaea, ASLO Archaea, Thermoplasmata, so Urea Archaeota. So that was interesting to see that in this near the shipwreck, we have much more diverse Archaea than away from the shipwreck. And similarly, looking at the bacterial core microbiome, which again, we use the 80% cutoff. So now we're looking at all the ASVs present in 80% of the samples, which was 640 samples for bacteria. And similarly to Archaea, there is a high diversity in the core microbiome, or the core members, near the shipwreck. And then as we go away from the shipwreck, that diversity of the core microbiome decreases and includes more cosmopolitan taxa. So it's again interesting to see that at that distance between 100 to 200 meters, we see unique core members that are not present anywhere after that distance, and some are only present in the samples that are collected immediately near the shipwreck. And as I mentioned in the methods, we used supervised learning, random forest models to see if we can predict certain distance groups based on the archaeal and bacterial ASV composition. And we found out that archaeal ASVs predicted proximity to the shipwreck quite strongly. And thanks to this analysis in CHIME2, we were able to identify specific ASVs that drive this prediction in certain distance groups. So we, for example, saw that Nitrosopumilis thamarchiota was a strong predictor for every distance, but for example, closest to the shipwrecks in this heat map, we can see that uncultured Bathyarchaea and Lokiarchaea were best predictors at the distances near the shipwreck. So that has that is related to what we saw in the core microbiome analysis. And what was also interesting is to see that distance groups that were best predicted from the ASV composition were the end members of the transects. So the closest to the shipwreck and the furthest away from the shipwreck. And for the bacteria, the prediction of the distance based on the ASVs was weaker than for Archaea, but we were able to identify specific ASVs driving the predictions in certain distance groups that also aligned with the ASVs that we saw in the core microbiome. And the distance groups that were best predicted by the bacteria were actually the 300-meter group and the 1,000-meter group. So to conclude this meta-analysis study, we saw that shipwrecks modify archaeal and bacterial sediment microbiomes in a sort of uniform and consistent way, where we did see elevated microbial diversity in the proximity to the shipwrecks that sort of trailed off or decreased with distance away. And we also saw distinct archaeal and bacterial core microbiome members surrounding the shipwrecks. And that was much more pronounced in the Archaea than the bacteria. And there's also a potential interesting metabolic shift in the communities surrounding these shipwrecks from more heterotrophic taxa like Bathyarchaea or Asgard-Archaeota to more chemolitho autotrophic taxa like Nitrosopumilis, which is the ammonia-oxidizing Archaea. So this is, of course, without doing the metagenomic analysis, but there's a potentially interesting thing to look at here in the future. And then using supervised learning, we made strong predictions of the proximity to these built habitats based on archaeal ASVs mostly, but also some bacterial ASVs. And it was really interesting to see that the strongest predictions were at the end members of the transect, so near and far from the shipwreck. So a couple of points I wanted to make here for future directions and what it all means for the global oceans. We know that these built environments, whether they're shipwrecks or oil rigs and other artificial structures on the seafloor, are distinguishable from the background marine sediments. And while these built environments are young in comparison to geological features like hydrothermal vents or seeps, we do see that they actually have some impact on the microbial communities in the ocean. And as we talk and think about this ocean sprawl that is leading to increased number of these habitats in the ocean, we sort of not really have much idea of how this is going to influence the ecosystem and microbial processes and things like microbial dispersal, genetic diversity, evolution, ecological connectivity, biogeochemical cycles, all these things in relations to build habitats and artificial structures on the seafloor are poorly understood. And one of the cool things I thought about this project was that we were actually able to make strong predictions based on predictions of distance from the structure based on microbial community structure. And maybe this could be used as a tool someday in detecting the influence of built habitats on the seafloor ecosystems. So quickly, I wanted to acknowledge the funding agencies that funded this research, which was NOAA and DARPA US agencies. Acknowledge archaeologists that worked with us on these projects, Melanie DeMore of BOEM, and of course the captain and crew of the research vessels Pointer and Pelican for taking us out on the water, and ROV, Global Explorer, and Odysseus field teams that helped sample and helped us use and navigate the ROV. And lastly, Hamden Lab and all the friends that went out with us to see and helped sample, specifically Rachel Magian and Urban Ray were also the members of the Hamden Lab. So thank you, and I am happy to take any questions.
So hello, and welcome to everyone who joined a bit later. Justyna, are you here? Yeah. Hi. Yes. This was a great talk, wonderful talk. Thank you very much. Now I would like to take 5 minutes for brief questions for Justyna. You can write them in the chat. You can raise your hand. We already have a first question. No? Okay. Yeah, there is a first question. Hans-Peter.
Yeah, thank you very much for this nice talk. Very interesting. Did you take hydrology into account? So did you see a pattern of the current around the shipwrecks?
Yes, we did take some measurements at least 2 of the shipwrecks, and because part of the project was looking at the dispersal of microbial taxa from the shipwreck into the water column, and we didn't really see any patterns when it came to currents. At one site, the currents were changing constantly, and there was no pattern with the dispersal. And the other side, we also didn't see much difference.
Yeah, I was just wondering, because you should have a lot of water mass transport around these shipwrecks. So the water is not stationary, and still you find these consistent patterns. So I'm just a bit surprised. And then I'm also having a question concerning the Nitrosopumilus. This is a kind of surprising for me, because it's known that it's adapted to oligotrophic conditions. Is there a substance which is released from the shipwrecks which might select for these?
We think that at least with the wooden shipwrecks that there might be a little bit more decomposition of organic matter, but we didn't really measure any total nitrogen or ammonium or nitrate or anything like that, unfortunately, for all these sites. So we think that Nitrosopumilus is just abundant kind of everywhere, in a smaller proportion around the shipwreck, and then increases in abundance going away from the shipwreck.
We also have a question from Everest. Would you like to read it yourself, or shall I read it from the comments? I will read it. Hi, Justyna. Oh,
Yes. Well,
great.
and if you want, you can read it. Doesn't matter. But if you prefer, I can read well, the question is basically if there is, if you expect more or less the same pattern of diversity in places where you are mining, for example, sand or things like this in the deep sea floor. If you mine sand, for example, you expect also to have more or less the same pattern with the diversity of bacteria or microbes.
Around the seafloor mining sites.
Yeah.
That's an interesting question. I'm not sure. I think the deep sea mining sites are more disruptive. You know, they're almost like a disturbance that's actively happening that may be affecting microbial community structure in a negative way versus these shipwrecks, you know, they land on the seafloor and then they stay there for hundreds of years. And maybe the microbial community sort of responds slower to that and adjusts with time versus the mining, I think, of the process as being more disruptive and actively changing the sediments around. So that would be interesting really to compare these influences. I'm sure there is an influence on microbial community from that, but I'm not sure if it would be the same as these stationary reefs are.
Okay, thank you.
Okay, if there are no more questions, then I would like to ask sort of a follow-up question. Oh, Emmanuel, okay, go first, then I will follow.
Yeah, thank you, Justyna. It was a great enlightening presentation from you. So, like Prof asked, that's Prof Peter, like I want to ask, what other factors may have increased the diversity of the microorganisms you were able to see in the location or in the site where the research was carried out outside?
What other factors?
Yeah, what other factors may have enhanced the diversity of the microorganisms?
Well, I think for some of these shipwrecks, it would be the decomposition of organic matter, like with the whale carcasses, for example, or woodfalls. There's plenty of studies on that. But on top of the actual wood decomposing on the seafloor, these shipwrecks are always covered, you know, with biofauna on them, like coral or barnacles, or there's just a lot of macrofaunal diversity around. So I assume maybe the decomposition of that would also affect these communities and sediments around the shipwrecks. And maybe that's why we sort of were hypothesizing that there would be a lot of taxa that are responsible for degrading organic matter around the shipwrecks and sort of then decrease as we go away from it. So we think of them as these hotspots of not just microbial diversity, but also macro diversity as well.
Roxanne, and then I will go.
Sorry about that. I guess I had a quick one for Justyna, and I might have missed it. So Justyna, I apologize if I did, but has there been work done on sort of the relative diversity as a function of shipwreck age?
There has not,
Okay.
as far as I know, and it's something that we wanted to include as well at some point in our analysis. But a lot of these shipwrecks are estimated from the 19th century. You know, it's hard to really tell how old they are. The steel shipwreck, one of the steel shipwrecks, we actually know what year it sank. It was 1945, and we didn't see that much difference between them. I think the size also matters. You know, we have shipwrecks that are 25 meters long, and we have shipwrecks that are much, much higher than that. So I think it's very complex and hard to parcel out other things or other influences.
Very nice work. Thank you very much for a really interesting talk, and to both speakers, actually beautiful talks. Thank you.
So Roxanne, thank you for mentioning first part of my question for Justyna, but the second part is not referring to the age, but do you have some idea about a difference in diversity when it comes to different type of material that these wrecks have, like metal and wood?
Yes, so we did compare. We did have 3 metal ones and 6 wooden ones, and then whole type, so whether it was metal or wood, was influencing the microbial community structure as well to some extent by the Permanova analysis, but not as much as distance, and water depth also was a significant factor, whether these shipwrecks were at 100 meters or 2,000 meters.
Thank you very much. And I would like again to thank both to Justyna and Joss for these wonderful presentations and nice discussion. And I would also like to remind all of you, those who were here from the beginning and those who joined a bit later, that if you would like to share, if you're an early career researcher and would like to share your work with us, please apply to be one of the speakers in our Amplifying Voices webinar series. And I hope to see you all and more on February 24th at the same time. Thank you very much.